Hello. In this short video, I'd like to show you some tricks for using the Gene Explorer in Bio 111. When you launch it, you'll see that there's a DNA sequence and a variety of pieces of information and some control buttons below. This DNA sequence may be different and the display may show slightly different RNA and protein structures, but the basic interface will be the same. The first thing you need to know is you can click in the DNA sequence, and by using the arrow keys you can move yourself around. The other thing you can do, and the most easy way to edit, is to simply type characters. If you type a lowercase character, like a lowercase g, it will change that base that you highlighted to a G. If you type a lowercase a, it would turn it to an A. If you type uppercase A, you can insert some A's, G, G, C, 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 and you can build up your DNA sequence that way. And this is really the best and most effective way to edit your DNA sequence as you like. I said, you click where you want to change, and then you can either insert by typing uppercase characters, or change bases by typing lowercase characters, and you can also delete by using the delete key. And again, you can move around using the arrow keys to different places. The other thing you'll want to notice is in this particular view, what you see is there's some DNA, and then in the section for pre-mRNA, there is none. So this section of DNA is incapable of making any RNA. And of course, because there's no mRNA, there is no protein being produced. That's what the none means, no protein being produced. So here now we have a little gene. And what you'll see is that you've got a promoter highlighted in green, a terminator highlighted in orange, and you make a little pre-mRNA, this little GGC, from that region in between. You'll notice that this GGC does not encode a protein. You'll see that you get a mature mRNA, GGC, with a poly A tail like you'd expect. And the protein is none, because again, this is incapable of making a protein. You can, of course, edit it to cause it to make a protein if you want. Now what you can see, now that I've edited the sequence, there's a significantly longer region that's made into RNA, and you get this longer RNA here, and you can see the protein that is produced. And these things align. So you see the AUG is the start codon, codes for the methionine, and then CCC proline, UU phenylalanine, UGG tryptophan are the proline, CCU, and then UAG the stop codon. So it makes this little protein sequence here. And it has, of course, its poly A tail. The blue is just for reference. It shows the protein expressed by the most recent version of the gene. You'll notice that this little gene does not have an intron, and sometimes we're going to ask for an intron. So, of course, you could go in and edit the DNA sequence to add the nucleotides to give yourself an intron. Now you see if I have edited this to include an intron. And what you can see is you have your DNA sequence, your promoter and terminator, your exons, and then, of course, an intron in the middle here. That's the exons are colored by these color scheme here. So there's a first exon, intron, exon. And you can see in the mature mRNA, it's now only the exons producing this particular protein. And you can scroll around and show, for example, here you are in the DNA, now we're in the intron, now we're moving to the exon. And what you see is that this base here corresponds to this base in the pre-mRNA, to this base in the mature RNA, and to part of that proline codon. And if I wanted to change that, I could type a lowercase g, for example, and then now it's we're now producing an arginine instead of a proline. So that should get you a long way to being able to do this. One other detail that you want to know, in case you need to know exactly which base you're on, you can see this down here, this selected base tells you which base you're on. So if you want to know what base you're changing or where you are in the gene, you can look at those numbers. That will help you. And occasionally this evaluate answer button will be useful for certain problems, especially on the SPOC.